What about societies, human societies and human culture? Anecdotally, I was asked in Hong Kong a few years ago by a woman who was teaching at a Canadian school. She was Canadian herself. What grade should I give my Korean students in the school, because if they don't get an A, their parents are going to beat them? Apparently in the Korean culture, it's acceptable if her observation is correct, and it seems to result in some kind of a motivation for doing well in school. And different cultures have different views about corporal punishment.
Well, there are kind of two questions, I guess, implicit, if I can. I may not be getting the drift of what you're saying. One, are there national differences in the prevalence of abuse, presumably representing culture, but not necessarily? And these data are hard to get, as I've tried to indicate, but at least the most recent international survey shows wide differences. But I can't say exactly where Korea fits into that. But the other question is, in a setting in which that kind of physical punishment, and again, it's a struggle to define where does physical punishment end and abuse begin, because when we're talking about child maltreatment, we're not talking about corporal punishment, which is presumably, as in the example you're giving, tied to a specific behavior in which the child can avoid that contingent by very clear family or cultural rules. There's a lot of debate, but when we're talking about severe maltreatment, we're talking about a child who is beaten to the point of being physically injured, and it's not meaningfully contingent on what the child does. It's clearly a product of an out-of-control parent, and there's no way a child can escape that. There's no rule by which a child can avoid it or end it. So you may be talking in Korea about corporal punishment. It's a big area of interest as to whether corporal punishment itself has some of these deleterious side effects. But most of the data we're talking about has to do with several levels beyond that. But the question still remains is, in a culture in which corporal punishment is widely understood as a legitimate and contingent response of a parent, would it have the same effect as in a culture in which that's nearly verboten? And it's a terrific question for which I don't know the answer, but maybe somebody else does.
May I add to that? There's an emerging field called cultural epigenetics. Cultural epigenetics, where it's a revolutionary field. These are usually cultural anthropologists who have learned a little bit about genes, and they have argued that different cultures have different distributions of particular genetic variants, probably as a consequence of several thousand or more years of selection, and that there are cultures where there's a bigger difference between high-ranking and low-ranking individuals, more emphasis on patterns of parenting, and the suggestion is that not only do these different cultures have slightly different genetic backgrounds, again as a result of where they've been living for the last several years, 1,000 years, but also in the pattern of epigenetic consequences of differences in how they rear their kids. And one of the most exciting areas that was recently published in Science, in the journal Science about 2 months ago, looked at China, where there is, I believe it's the Yangtze River that roughly divides China in half to northern China and southern China. For thousands of years, those living in northern China have been raising farming wheat. Those living in southern China have been largely raising and harvesting rice, and their social patterns in the two groups are completely different, as are the attitudes of the individuals and as is the way they rear their kids. So here's an example of where it's a chicken and egg thing. What came first? The bottom line is that they have very different ways of living socially in terms of either working in a community fashion as opposed to working more independently. And it has consequences with each change of generation.
My question for Dr. Rees is three-pronged, but I promise each point is brief, so I just want to give these to you in a chunk to think about.
Okay.
You
I appreciate
presented-
that.
You presented some methylation data on gene methylation, but you didn't say which one. I was just curious if that is known. That's one piece of the question.
Okay, can I answer that first?
And answer what's methylation, because not
The
everybody
methylation
here
is that CH3 group that he was
Okay.
talking about that silences the gene.
Okay, so you take a tiny molecule that consists of 1 carbon and 3 hydrogen,
Stick it on a gene.
and you stick it on a gene in or near a promoter.
No, but you showed that endogenously in some of
Right,
your,
right.
in your human.
So the specific gene involved in the data I showed was the world's most famous gene. It's called the serotonin transporter gene. Those of you who, as Kate, who's a neuroscientist, know, it's the world's most studied gene because of our report in 2003 that gene has 2 different forms, a short and a long form, and the short form in combination with external stress makes young adults more vulnerable to depression subsequent to the period of the stress. So there's a lot of debate as to whether that is replicated or not, but neuroscientists worldwide took a great interest in that, and that was the gene that was methylated. But as Steve's data show, and I think we can say pretty confidently, if you've got a strong environmental influence, adverse or maybe even positive, is in terms of the treatment, and there's some human treatment data that map onto Steve's data, I might add. We now know that it's likely that hundreds of genes are going to be infected, and probably in the study I showed you, that's the one gene they looked at because everybody in the world wants to know about that gene, but there are probably 100 or 2 others who are also involved.
The second most studied gene is the glucocorticoid receptor gene, and that also has a polymorphism. And that was the original Michael Meany-Moshe-Zipf gene.
Will you yield?
Yes, I will absolutely yield.
Okay. Bill?
You talked about the young children raised in dire circumstances in Romania, and some of them went over to the UK. I'm just wondering if there's a study of the reverse, where you have children raised in healthy circumstances who later on in life become unfortunate and fall into abusive situations. Does their healthy initial years provide any kind of genetic immunization against later adversity?
Can I not answer that question, but try to provide something of interest anyways? We have 2 French studies in which we have children of parents with low education and presumably lower IQ, raised by families very well educated and presumably with higher IQ, and every combination, high, low, and high-low. And what we know from those studies is that the advantaged environment just about makes up for the presumed genetic disadvantage. So that's suggesting the power of a positive environment. But even among those with very fortunate rearing environments, there's a clear difference between those born from parents with low education and presumably low IQ and those high. So the rank order remains the same, but all boats are lifted. But that doesn't quite answer your question, because that's genes, that's not environment. Do you know of a study in which has tested whether a fortunate early environment protects against later adversity?
Not an experimental study, but there are occasions in the wild where sometimes growing up in a very fortunate circumstance puts you at risk when you get into trouble later because you don't know you haven't learned how to cope.
Yes, ma'am, the first row.
I think you'll be able to answer this. It's a birth weight question in a Jewish context. So, given the restriction on maternal calories that would have preceded these births, have there been any studies of children born in DP camps?
Oh, yes. Well, the absolute most informative studies are children born during what's called the Dutch winter famine. There was a period just towards
1940.
the end of World War II, or what I'm going to call now World War III, in which, and this happened tragically all over Europe, but in the Netherlands, the Dutch thought that the Nazis were finished a few months before the Nazis actually were finished, and they rose up in preparation for the Allied liberation, and they rose up too soon, and the Nazis, instead of conserving their resources, literally starved about 1/4 of the Netherlands for about a 6-month period, in which those kids were born at extremely low birth weight, but that only lasted 4 months. Then the Allies did come, and those children rapidly returned to the normal growth level, but those kids have had trouble lifelong, of just the kind I'm talking about.
And still have their kids.
Yeah.
Dr. Tsunami, you mentioned about the males in the colony, as they become of age, they leave and they try to then rejoin other groups. And sometimes their behavior, they get pushed back out again. Do they ever seem to learn and modify their behavior, or are they very locked into just behaving a certain way?
Depends on the male. There are huge individual differences, and they have different strategies for getting into a new group. And it basically has to do with being able to learn to be submissive under appropriate circumstances rather than trying to fight your way into the middle, which will get you killed if you're a rhesus monkey. To be patient and to be resourceful. It has a great deal to do with overall social competence. And those males who are well socialized, play a lot, have a lot of friends who are peers, usually can make that transition easier than ones who have been ruling the roost because their mothers are high ranking and they don't know how to be nice.
Mark?
So again, social capabilities is the big thing, and that's what all of this early experience is about. It's a socialization process. It's learning what the rules are, learning how to judge where in the hierarchy you are, what you can get away with, and what you have to be careful about.
You gave a very interesting and hopeful, encouraging example of intervention through foster parenting, of reversal of environmentally artificially created changes in brain chemistry caused by peer rearing. Then you mentioned, but you didn't give us an example, that you say that you have demonstrated, if I understood you correctly, that you can change the dominance package in the brain chemistry. I'm just wondering how you do that. What kind of intervention is involved?
You can take a high-ranking individual and put him or her into a new social group, and suddenly that rank changes dramatically.
In the new group or the old group?
Both, actually. In the old group, if you remove a high-ranking individual, somebody else is going to move up, and there'll be a rearrangement. This happens in nature, not every year, but occasionally, and when it does, the consequences are enormous.
Does this affect the brain chemistry?
As near as we can tell.
Not only the brain chemistry, but the placental chemistry. That's a stunning finding, that the placenta, just for you guys here, the placenta is a fetal organ. It's not the mother's organ. It's the fetal organ. So you've got fetal tissue. You can tell the social stratum of the mother by the gene expression profile in fetal tissue.
That's a big deal.
Right? So, I mean, you just heard, I mean, you don't get findings of greater importance than that in our world than you just heard from Steve.
How many troops do you able to keep at that in Poolesville?
Pardon me?
How many troops do you have there?
We have one group that lives in that 5-acre enclosure that's about the size of a mid-size wild troop. And then we have smaller social groups, a larger number that live in expansive indoor-outdoor pens where we can manipulate both the genetic and the early experience background of them. So the troop in the field station is our best effort to recreate what you see in the wild and to look at the kind of biological measures and gene expression measures that we see that would be comparable to what you could get in the wild, and Zalman's hierarchies and all that, but also smaller groups that we can manipulate, we can cross-foster, we can differentially rear, and then we have a basis for comparison in terms of, say, levels of cortisol or levels of serotonin metabolite with what is actually naturally occurring. So we basically go back and forth between smaller groups that we can control experimentally and naturalistic groups. We can observe a phenomenon in the naturalistic groups, see if we understand what's going on by careful experimentation, get the results of our experimentation, go back into the wild or into our natural group and see if we have a naturally occurring counterpart to which we have been able to manipulate and understand. So it's a interactive process. It's an iterating process where we go between carefully controlled laboratory studies and much more naturalistic situations, and we have the luxury of having access to 2 field sites where it is actually nature.
You mentioned depression, and I was wondering if you ever noticed signs of other forms of mental illness like bipolar disease or even schizophrenia, and if you have, can you trace that genetically or mitigate it by moving monkeys that have those symptoms into a different cultural, higher or lower level?
Okay, I assume you're asking me.
Yeah.
So the things I emphasized or alluded to, say anxiety/depression, I said it's about 20% of the population in the wild that are prone to that, and in our lab, and maybe this very impulsive aggressiveness, maybe 5 to 10% of the population. And both of those numbers are somewhat similar to my best reading of the human literature as to the incidence, at least in Western society. Bipolar or schizophrenia is much, much less frequent in humans, and we have interventions, psychiatric interventions, medical interventions, that allow these people to survive in most cases. I don't think if there is a natural component to that, natural counterpart to that in monkeys, you're looking at a very small part of a population. We don't have thousands of monkeys to look at except maybe at these field sites. And my guess is any individual so afflicted would not survive to reproductive maturity. That is, that individual would not be able to live for extended periods of time, long enough to produce offspring of their own. I have asked my colleagues who have extensive field studies, if they've ever seen anything like that. Characters, let's say, take schizophrenia. Individuals who, when they're growing up, seem to be socially listless and not as competent as their social background would suggest. And then when they hit puberty, they start doing really crazy things. And most of those animals are dead within a couple of months because they do really crazy things.
How do they die?
They get into unnecessary fights, they take unnecessary chances, they get socially excluded and picked off by predators. So over the generations of humans, we've learned sort of how to deal with that. Mother Nature isn't so kind.
This isn't scientific. Given the importance of the work of both of you and the findings and the fact that you're probably very far from being finished at this point, what have you done to see that your work is going to continue after you can't do it anymore?
Funny you should ask that. NIH, I'll speak for myself, NIH has what they call forward-looking plans, or where do you want to be 5 or 10 or 15 years from now. I don't think we've been working together for 4 decades, or I was very young when we first had our contact. I'm not that old, but I'm not that young either. And I want to keep, the thought of retirement has never entered my mind, but it might at some point. At the very least, because I'm a federal employee, I don't have to retire. I could keep running a lab until I was on my deathbed, which is what my father actually did. To the great benefit of the world, I may add. But I think at some point, it's time to turn the day-to-day responsibility over to other individuals, and I've been grooming them very carefully. And at that point, the NIH has recently instituted an emeritus program where you can, quote, retire, be on half-time, and 20% of your half-time has to be explicitly directed towards mentoring. I've been 30 years away from teaching introductory psychology classes, which I love teaching, and I like mentoring, and that would be a chance to make it official. But yeah, we're grooming successors for sure.
I'm a little more pessimistic, and let me, since we're talking about a process in science, and this is a chance to talk about what really goes on and what the prospects, but not actuality, of Tikkun Olam in science funding. I think there's good reason to be worried that work like this will never produce the significant effects it has great promise of producing. One is, for the last 3 years, are the only 3 years in the post-war history of the National Institute of Health in which the actual budget has gone down. Now, there are periods of time in which it stayed relatively level, which is functionally reduced because research costs more and more. But now the funding is going down. And what it means is that from institute to institute, only 6 to 7% of grants that are reviewed, which is about half the grants submitted, are being funded. Now, Dr. Bennett runs a review committee, but she will tell you, I am sure, that it's impossible for human judges of science to reliably pick the best 3 to 4% of applications when a large number of them are competently done. I can also tell you, and Dr. Bennett can confirm this, that the preponderance of funding is still going to older and not younger investigators. So younger investigators, although there are many, many programs to encourage them, even in an elite university such as I work in, younger investigators are regularly getting discouraged. There's a third factor that I think it's very important for the public to understand, which is there is a trend in science funding to latch on to hot topics. And the combination of hot topics and low funding is disastrous if you happen to be in a topic called cold. And at the moment, the very hot topic in the National Institute of Health is called personalized medicine. President Obama advertised it in his State of the Union. It is the calling card of Francis Collins, who is an estimable leader and a brilliant scientist himself and has played a huge role in decoding the human genome, and that work was crucially important to all the gene expression work we're talking about here. But he has started an initiative called precision medicine, which means each patient gets exactly the treatment that their particular profile requires. I can tell you that a history of child maltreatment is 100 miles away from what Dr. Collins has in mind for precision medicine, although many people would argue that if you were picking 10 facts about a patient, whether or not they have been maltreated is crucial for tailoring a treatment program. So we're headed for probably more cuts. And what's crucial for everybody in this room to try to engage in is trying to separate the politics that opposes big government for the politics that understands the role of government in healthcare. And we in healthcare have to understand the importance of giving primary support to young investigators and for freeing scientific review from an impossible job of discriminating the top 3 or 4% of applications. So this is a citizen's job, and everybody in this room has got to recognize that research of this kind is deeply threatened by what's going on. You don't have to be a Democrat. You don't have to be a Democrat. But what the position you have to take is when we're talking about funding research of this kind, it's not going to be done by industry. It is not going to be done by industry. It's a government function. And NIH, with a few small warts, is a government agency you as a taxpayer can really be proud of. But you've got to defend it, and you've got to clarify the language to understand that this is not overwhelming big government. This is a function of any national government, and European governments understand this. Even David Cameron protected his scientific funding when he went through all the crazy cuts in social spending. David Cameron and the Conservative government in the UK protected funding. It's going up, ours is going down. I can tell you the health of Britons is already substantially better than the health of Americans, class by class. And we're really going to be in the backwater if we don't straighten that problem out. And that's got nothing to do with child maltreatment. It has to do with the maltreatment of the entire country.
But I wanted to kind of come back and maybe come full circle to the whole theme of this. Remember, there's a biblical saying about the sins of the parents shall be visited on the children to the 7th generation. Sure. And I think some of what if I got that quote right?
4th.
How many? 11th?
4th generation.
3rd and 4th.
But in a what?
Anyways, it's a number of generations.
Yeah, and in a way, you're showing that the treatment is actually being locked into the genome, and it's somewhat carrying it on.
So one of the goals of my research is to take early adversity that does cross generations and somehow break that cycle.
If I, as a concerned citizen, concerned about eliminating cuts, I mean, in the whole sequestration, I was going to say idiocy, but or I'll say process. If I wanted to write an informed letter to my congressman or to my senator, is there, I haven't been taking notes, and I understand the general gist, but I would like to express my concerns in a letter that reflected what you've been saying. Is there some text I can refer to, since I didn't take notes?
You mean you'd like to cite what you've heard?
Yes.
Yeah.
If I wanted to write to Van Hollen.
An easy, aside from Steve's, and to a much lesser extent, my publications, which are all available online.
Well, okay, in that case,
Yeah.
I'll be able to.
Yeah, but the initiative we're talking about has a nickname. It's called reversibility. And it refers to the reversibility of the liability for illness in adults incurred by childhood adversity. Reversibility is the single term. If you just put in,
But use that as a key word.
if you put reversibility NIH,
Okay,
you will have about 10 or 12 web pages. And if you wanted to write and say, here's an example of a project A, which is just getting off the ground, B is severely threatened, and C is being better funded in Europe than in America, particularly in the UK, you'll have plenty of ammunition. But just make sure, make sure you're writing. Don't preach to the converted.
well, that's why.
Just find some suitable. Those of us who live in D.C., of course, don't have anybody to write to, but find.
I live in Bethesda, but I can't write.
But try to find, or if you're a member of a professional organization, or if you're a member of a congregation. I personally think that every Jewish congregation in this area ought to take this really, really seriously and take a congregational position and write their representatives or senators, and even if they're favorable, to give them a way of thinking about defending the NIH budget. It just couldn't be more important.
I would never have heard of this fascinating topic were I not connected with the Institute for Science of Judaism. To what extent have you publicized your work in popular venues, like popular journals, television programs? It's fascinating stuff, and most people don't have the foggiest notion of what's been going on.
Well, Steve, he can tell you, Steve's work is very widely known. Articles about his work have been appeared in many major general journals, newspapers. Steve he may not tell you this, but he's been invited several times to provide information to the British Parliament, Britain again. His work is very widely known. The center of this new initiative on the reversibility, or the potential reversibility of these long-term effects, it really is an important judgment call when you want to go public. I mean, we're obviously going public tonight, but it's very different because we have a chance to clarify what's known and unknown, and you have a chance to ask questions. So the opportunity for misunderstanding is very much minimized in this kind of more intimate setting. But when do you start going public with major scientific findings is a big question. And I don't have the answer to that. But my own sense is on the very specific topic of adults at risk for illness and the extent to which we can do something about it, which is what the public would like to know, I would caution my colleagues, this isn't quite the time to go public. And I've spent, since I work in genetics primarily, and I haven't talked much about that, the field of genetics is almost ruined by the premature publication of findings that either don't hold up or are badly misinterpreted. We have a phenomenal example of that in the New York Times of 2 weeks ago, where a happiness gene got front page on the News of the Week. I mean, it was so premature, and there's no replication. So in terms of the main topic that we're trying to assemble a sensible program of research for, I'd be uncomfortable, but my colleagues and I would differ. I'd be uncomfortable on that topic to go forward. Steve's findings, his major findings, have been replicated in many labs. They have a solidity to them that deserve very wide attention. My part of the talk, I'm happy to share. I want to underscore the uncertainties about it. I'm a little hesitant to move it out further at this point because there's some potential harm, and people have had maltreatment in their past, many people, adults. We don't know exactly which people who have been maltreated are at risk and which aren't. I'm a little hesitant to go further with that. But Steve, I'm sure you've got very different views on that.
Not quite ready for prime time, I think is the way to put it.
Right.
But practicing a lot.
I would add to your comment about hot topics that I think you're onto something with the epigenetics. I do sit in a position to see what's coming down the pike, so epigenetics is hot. And I thought you're, again...
Epigenetics is hot, child maltreatment isn't.
You've got the in on epigenetics, but regardless, your data are beautiful and interesting. I was remembering the regression curve that you had with the many dots, and the data, I think you would agree, are a bit noisy. There's still a nice regression there. But I was wondering about the confounds between the environment of the specific child having their genome epigenetically altered versus something that you mentioned, that they're probably dealing with a parent that's got mental illness, addiction, additional problems. There's also this intergenerational transmission. And I'm wondering if you start teasing apart that, if that regression, your R values might improve. And I'm thinking of a couple of examples. It'd be hard to find, but there have been some lay books. Look Me in the Eye, Asperger kid was grossly mistreated. He swore up and down when he had a child he was going to treat that child great. So there are a few examples. A child called it grossly mistreated, and then had a child and bent over backwards. So I'm wondering if you're making some effort to tease apart those twin issues of the specific trauma in childhood versus something that might be generationally transmitted, these methylation patterns based on a parent that's also suffering.
Well, somebody mentioned the twin studies, and twin studies have, my answer was a little too quick. They have struggled with twin studies, and twin studies would give you a bit of insight into that. The most crucial human experiment is an adoption study, because you can ask, is it the environment in which the child is reared, or is it the characteristics of the parent? So that would help a great deal. Treatment studies would give you some help if you could reverse the specific defects with a social treatment. Might give you some sense as to the potency of the social environment. That's the only experiment we can really do in humans. But it's not an easy distinction, because although adoption agencies are not very good at screening adoptive parents, and they have the same, for example, divorce rates that the normal population does, and the same psychopathologies as a normal population do. Adoptive parents, by and large, don't abuse their kids. It's pretty rare. So you don't have a good environmental opportunity to separate severe abuse as an experience versus the consequences of the genetic liabilities that an abusing parent faces on. And I think in the end, I think the partnership that what Steve has done for this whole initiative is help us clarify. I hope you get the picture in Steve's work. It's a reciprocal process. He sees a finding in the human literature and he tests it, and then we take his findings and make effective use of it the next step. I think that I would have to say that David Barker's work, and that's why I keep coming back to that, it's a paradigm. We could not really get that clear until we could absolutely control fetal growth we didn't tie off, we collectively didn't tie off the uterine arteries. We narrowed them just enough to produce exact gradients of rodent newborn weight, that clinched it. That really, really clinched it. It was fetal nutrition. All the reasons that a mother isn't adequately nourished could have been transmitted, but the fact that it was malnourishment that was the causal factor got nailed down by animals. So that's, I think we can try with various human designs, but I think there's too many compromises.